The overall goal of this microscopy-based approach is to observe essential processes during growth of bacterial cells.
This method enables us to address important questions in bacteriology, such as how essential processes are coordinated during bacterial cell growth and division. The main advantage of this technique is we're able to use fluorescent reagents to visualize bacterial cell surfaces and structures in living cells.
Here, we use this method to provide insight into essential processes in Agrobacterium tumefaciens. However, this process could also be adapted to other bacteria to study their growth as well.
To begin, use a sterile wooden stick or pipette tip to inoculate 3 milliliters of ATGN growth medium with a single colony of a wild type or mutant strain of A. tumefaciens. Grow the culture at 28 degrees Celsius and 225 RPM overnight. The following day, use a spectrophotometer to measure the OD600. Then, dilute the cell culture to an OD600 of approximately 0.2 and continue to grow the cells to an OD600 of 0.6. Dispense 1 milliliter of exponential culture into 3 microfuge tubes and pellet the cells in a desktop centrifuge at 7,000 times G for 5 minutes. Resuspend each cell pellet in 1 milliliter of PBS and then add either 1 microliter of DMSO orange stock solution or DAPI stock solution. Mix gently by pipetting and incubate the resuspended cells in the dark for 5 minutes. Pellet the cells by centrifugation at 7,000 times G for 5 minutes and resuspend the pellet in 1 milliliter of PBS to remove excess reagent.